Hi, folks. So in this lesson, we are going to go over standard 3.2.2 about offspring ratios. This is the 18th standard in our biology class, and it falls in our 7th unit about genes and inheritance. So this standard says that students will be able to predict offspring ratios based on a variety of inheritance patterns, including dominance, codominance, incomplete dominance, multiple alleles, and sex-linked traits. So while that's the language of the exact standard, we want to walk through what this might look like for you all as you approach your EOC exam. So in this video, this first video, we're going to break down the standard. We're going to talk about an introduction to genetics. We're going to introduce the concept of Punnett squares, and we're going to walk through ratios as well. I just want to point out, for those of you who may not be familiar with our style of presentation, when you see a highlighted green section of text, you can pause there to read the Spanish definitions or to read the Spanish translation. It will be the same slide from the previous portion of the video. So as a brief introduction to genetics, genetics is defined as a branch of biology concerned specifically with the study of genes, genetic variation, and heredity in organisms. So as we can see in this cute image of these young people, these young children, these babies, genes allow us to have a great deal of diversity, a great deal of variation. They are what give us all of our difference, not only human beings, but all forms of life on our planet. So some important terms to know as it relates to genetics. Phenotype. A phenotype is a physical description of a trait. So this might, for example, be black hair. An allele is one of two, could be more, variations of a gene. In this class, we will typically be working with genes that only have two variations, so there will only be two alleles. A single allele will be represented by a single letter. All of these definitions will be available as we go through them, and you'll be able to pause the video and write them down. A genotype, instead of being a physical description, this is the genetic description of a trait. This is a person's two alleles. Each person gets one allele from each of their parents. We get one copy from each of our parents. So for example, we might have two alleles that are different size, capital and lowercase, capital B, lowercase b. We'll talk about that shortly. We can describe genotypes as being one of three things. They could be homozygous dominant, which is a genotype that consists of two dominant alleles, often represented by two capital letters, for example, capital K, capital K. We could describe genotypes as being heterozygous. This means that a genotype is consistent of one dominant allele and one recessive allele. So this would be represented often by a capital letter next to a lowercase letter, capital K, lowercase k. Now we can also describe genotypes as being homozygous recessive. So this is a genotype that consists of two recessive alleles, often represented by two lowercase letters. Now, we want to be aware of the fact that the prefix homo means same. So if I'm homo-dominant, then that means I have two of the same alleles and they are both the dominant allele. Homo-recessive means I have two of the same allele, but they are both recessive. They're both lowercase, and then hetero as a prefix means different. So that means that my two alleles are different. So we should be pretty familiar with this image. You all have seen similar images in our biology course before. But when we have two sexually reproducing organisms, they mate, the male sperm fertilizes the female's egg, as shown in this image on the left. Their unique genomes combine to form a unique offspring. For a number of reasons, we have an investment. We want to know what to expect from this fertilization. And we can reasonably predict the likelihood of an offspring having certain traits by using what we refer to as Punnett squares. And this is going to be super helpful to us, especially as we think about the biotechnological implications. We might want to know how to prevent a baby from having specific diseases. We might want to know the likelihood that a baby has specific physical traits. Punnett squares allow us to do just that. So you might be wondering at this point what exactly a Punnett square is. You've heard this term tossed around by your biology teachers. You maybe have heard it tossed around in the documentary that we watched a few weeks ago, but you maybe don't know exactly what a Punnett square is. A Punnett square is a square diagram that is used to predict the genotypes of a particular cross or breeding experiment. We use it to determine the probability of an offspring having a particular genotype. So there's a lot there. If you want to pause the video and read it to yourself, that might be a good idea. But let's see exactly what these Punnett squares can be useful for. So what we have here is a Punnett square. It's a masterpiece, right? It's a very simple diagram. It just has four squares right next to each other. It's almost like a chart of sorts. And we're going to really walk through how you can use these Punnett squares to discover the probability of an offspring having a specific trait. So here's our question. Detached earlobes are dominant to attached earlobes. A female with attached earlobes mates with a heterozygous male. What are the chances their offspring has attached earlobes? So at this point, we may not have any clue where to start, but if you've been attending our live lessons, then you definitely know that a key is always a great place to start. So you're going to see us start to develop this key. You might be wondering what goes inside of a key. You should first determine what your alleles will be. Obviously, you have a dominant allele and you have a recessive allele. The problem tells us that detached earlobes are dominant to attached earlobes. So that means that our dominant allele represents detached earlobes. So that means that that allele should get the capital letter. It could be any letter. The letter itself doesn't matter. If you want to make things easier on yourself, you can use the first letter of your first name or whatever your favorite of the 26 letters in the alphabet is. But because we've got earlobes, I'm going to work with the letter E in this question. So the dominant allele is represented by a capital E. The recessive allele is represented by a lowercase e. But we still have some more information that we need to represent in our key. And we develop these keys because they'll be really helpful to us as we start to fill in information in our Punnett square, and then as we start to try to answer the question. So we also need to figure out what the three possible genotypes will be. In this case, and in all cases, those three possible genotypes will be homozygous dominant, heterozygous, and homozygous recessive. So my homozygous dominant genotype has the same two alleles, that's what homo means, same, and they are both dominant. So that means I need two capital E's. Hetero as a prefix means different, so heterozygous should have one of each, one dominant allele and one recessive allele, one capital E and one lowercase e. And my homozygous recessive, homo of course means same, but this time I want the recessive alleles, so two lowercase e's, two recessive alleles. Great. Now this is going to be very helpful to us because of the next sentence in our question. It says a female with attached earlobes mates with a heterozygous male. Okay, so let's walk through that a little bit. In order for a organism in this case to have attached earlobes, that must mean that they are homozygous recessive. How do I know that? I know that because it says that detached earlobes are dominant to attached earlobes. That means that anytime there is a dominant allele present, that dominant allele is going to take over for the recessive allele. The dominant allele in that case will determine the trait that the person has. So if a person has detached earlobes, they must have a dominant allele present. But our female here has attached earlobes, which means she does not have the dominant allele. She must be homozygous recessive. So now that we know the female's genotype, we've discovered that she is homozygous recessive. Let's talk about the male. Well, that one's pretty easy. because the question tells us that the male's genotype is heterozygous. Of course, this means that he also has detached earlobes. We see that a heterozygous genotype has a dominant allele present. It's got a capital E. And so that capital E takes over. It tells the little e, your services aren't needed. I'm going to determine what the trait is for this person. And because I am the dominant allele, that means that this person has detached earlobe. That's the trait that I'm giving them. So now we know what each of these two individuals' genotypes are, and we also know their phenotype. So we need to start working within our Punnett square. It doesn't matter where you put the male or the female. It doesn't matter where you put each organism. I'm going to put the male on top and the female on the left in this case, but you could go vice versa. The female's genotype, of course, is homozygous recessive, so she gets two lowercase e's, and the male is heterozygous, so he gets a capital E and a lowercase e. Now, these Punnett squares work pretty simply, but I'm going to represent it with arrows so you can see exactly what happens. I'm going to start off in the box in the top left. In that box, I'm going to drop the capital E down, and I'm going to swing the female's lowercase e over, just like that. So my arrows are representing the movement of these alleles. That's ultimately what they are. We refer to them as letters, but they're really representing these alleles. So in this top left box, I end up with a heterozygous genotype, one capital E and one lowercase e. Next, I'm going to move to the box in the top right. I've got a lowercase e from the male that's going to drop down, a recessive allele, and I've got a lowercase e from the female that's going to swing over, also a recessive allele. So it's going to look like that. This gives me a genotype that is homozygous recessive. Same, homo meaning same, and recessive meaning lowercase. So at this point, hopefully you're getting the swing of things. I'm going to drop down to the bottom left box. My capital E from the male, which is a dominant allele, is going to drop down. And my lowercase e from the female, this time the other lowercase e, the recessive allele, is going to swing over. Again, I end up with a heterozygous genotype. So I know you guys are smart people. You're picking up on what's happening here. I'm going to go ahead and finish out my last box. My arrows show me the movement of these alleles. And again, I end up with a homozygous recessive genotype. Now that I've got my Punnett square filled out, I can actually begin the process of analyzing these genotypes so that I can answer the question. Well, what I see in these genotypes is that 2 out of 4 possible offspring are heterozygous. So they have the detached earlobes. And 2 out of 4 possible offspring are homozygous recessive. So they have the attached earlobes. To answer the question, which we'll revisit, the question is, what are the chances their offspring has attached earlobes? So if 2 out of 4 possible have attached earlobes, then there is a 50% chance of the offspring having attached earlobes. I'm going to pause here so you can take in all of this beautiful biology and math on your screen. You can pause it, you can write it down, and I would suggest you do that. We do have a practice question coming up, and that will be an opportunity for you to pause the video and try one on your own. Great. So a Punnett square is what is really giving you a ratio. So you might be wondering, what is a ratio? Maybe you are in math 2 right now and you don't fully understand this concept of ratios yet. There's our Punnett square. We've obviously got 4 boxes, and those 4 boxes represent 4 possible outcomes once I do my cross. And a cross is just showing how 2 organisms mate. So I've got 4 possible outcomes, 1, 2, 3, 4. So these 4 possible outcomes represent a 25% chance of having an offspring with that ratio. So box 1 is a 25% chance. Box 2 is a 25% chance. Box 3 is a 25% chance. And then obviously box 4 is also a 25% chance. So that's what allows us to work through probabilities. Probabilities tell us the chance that something takes place. So let's just say that two of these boxes have the same genotype, box 2 and box 3. They have the same genotype. A third box has another genotype, which I'm going to represent with blue in box 1. And then the fourth box has yet another genotype, which I'll represent with green. So we've got 3 possible genotypes that have come up in our Punnett squares, which you all are likely to see. Let's determine what the ratio of these genotypes would be in this case. If my red genotype, my genotype from box 2 and 3 is going to be first, well, then I've got 2 of those boxes, 2 out of the 4. Then I've got another genotype in box 1. So there's only 1 of those genotypes represented. And then in box 4, I also have just 1 of those genotypes represented. So my ratio is therefore 2 to 1 to 1. And the percent chance of having the first genotype is 50%. The percent chance of having that red genotype represented in boxes 2 and 3 is 50%. Why? Because 2 out of the 4 boxes, 2 divided by 4 is 0.5, 50%. 2 out of the 4 boxes are going to give us that genotype. So this is how we can determine ratios and probabilities from Punnett squares. We can get both of them. You just need to pay close attention to what the question is asking you. So at this point, I want you to pause the video and try a practice question on your own. The question is, in a particular strand of beans, green pods are dominant to yellow pods. A farmer mates a stalk of homozygous dominant beans with a stalk of yellow beans. The farmer expects to yield, which means to produce, 100 stalks. How many stalks with yellow beans should they expect? At this point, pause the video, work on the practice problem, and the answer will be available to you in 3, 2, 1. You need to make your key per usual. This is what a key would look like if I were making it. I represent my alleles. I represent my genotypes. My homozygous recessive genotype is the only genotype that gives me yellow beans. So when I do my Punnett square, I have a homozygous dominant genotype that represents the green bean. And then I've got my homozygous recessive genotype that represents the stalk of yellow beans. I do my cross and I come up with all of the resulting offspring being heterozygous. All of them are heterozygous. This tells me that my answer is this farmer or these farmers should expect 0 stalks with yellow beans. 0 of their 100 stalks should be yellow beans. So in review, we covered standard 3.2.2, which was about offspring ratios. We defined key terms about genetics. We introduced the usefulness of Punnett squares. We practiced making a Punnett square and answering a genetics problem. And we explained ratios and probability. So in this video, we talked exclusively about complete dominance. In the next video, come back and check us out so we can talk more about incomplete dominance, codominance, multiple alleles, and a few other topics as well.